Hello, everybody, and welcome to our Precisionary Webinar. Today is August 22nd, 2023, and we are really honored today to host Dr. Omolara Baiyegunhi, who is a research associate and Wellcome Trust Early Career Fellow. She is located at the African Health Institute, Research Institute in Durban, South Africa. So she's joining us with quite a few hours of time difference. And thank you so much for being here. She was a postdoc fellow from October 2018 to January 2023 and was an honorary research fellow at the University of College London in the UK for the several years after. Dr. Baiyegunhi received her Ph.D. in immunology in 2018, and in her current research, she aims to identify the virological and metabolic factors that influence the development and persistence of HIV-1 reservoirs. And Dr. Baiyegunhi is passionate about her excellence in scientific research, and we are honored to host her. So today, she is going to discuss and help us understand the impact of the timing of antiretroviral therapy initiation on T follicular helper cell responses in HIV patients, and explore the persistence of HIV antigens in lymph nodes and its implications for disease progression. And also she'll help us examine the role of HIV-specific CD8 T cells in clearing antigens from lymph node tissue sanctuary sites and its potential therapeutic significance. So with that, Dr. Baiyegunhi, I want to hand it over to you. And again, thank you for your time and your talk today.
Okay. Hi, everyone. Thank you, Abby, for the kind introduction. Can you confirm that you can hear me?
Yes, we can hear you and also see your presentation.
Okay, great. So, as Abby mentioned, I work at the Africa Health Research Institute, where I am a Wellcome Trust Early Career Fellow and Research Associate. And today I'll be talking to you about HIV antigen persistence in lymph node T follicular helper cells and how this is mitigated by functional virus-specific responses during hyperacute treated HIV-1 infection. So here is an outline of my presentation today. I'll firstly focus on the effect of the timing of antiretroviral therapy initiation on the development of T follicular helper cell responses. And then I'll move on to talk about HIV antigen persistence in the lymph nodes of individuals that started antiretroviral therapy very early in acute HIV-1 infection. And then I'll also offer some insights into the mechanisms that we think are contributing to HIV-1 persistence or clearance within the lymph node tissue sanctuary sites. So as a bit of introduction, I would like to highlight that antiretroviral therapy has drastically decreased HIV-related mortality. So this has changed HIV from death sentence even to a chronic manageable condition. However, the HIV epidemic still remains a global health challenge. And here in this slide, we can appreciate that more than 38 million people are living with HIV all over the world. And more than 50% of these people are residing in Eastern and Southern Africa. So this highlights the importance of an HIV cure or of novel interventions that can keep people off long-term antiretroviral therapy. It's important to note that a cure or sustained HIV-1 remission in the absence of antiretroviral therapy is possible. Although for now, it's still a pipe dream, because what actually happens is once individuals stop taking antiretroviral therapy, you can see individuals on antiretroviral therapy have very suppressed viremia. But once they stop taking antiretroviral therapy, you have this rebound of viremia. And this typically occurs for most individuals. But then we have like special individuals that are able to control HIV in the absence of antiretroviral therapy. And there have been efforts by scientists over the years to try to initiate an HIV cure. And in one such studies, which we call the Boston patients, where these individuals underwent stem cell transfer to actually try to cure them of HIV. And the sad news is that after 3 and 8 months of being off antiretroviral therapy, there was rebound viremia. But it's not all bad news, because we have other cases where there have been sustained suppression of HIV viremia in individuals that have stopped taking antiretroviral therapy. But the challenge is that these individuals had to undergo stem cell therapy, which was used to cure leukemia or Hodgkin's lymphoma or any such cancerous disease. And as we can appreciate, it's not a viable pathway to a HIV cure. I just want to highlight some of the patients, the most prominent being Mr. Timothy Brown, who unfortunately demised due to his leukemia. But before his demise, he had been fully suppressed of HIV viremia, even after his stem cell therapy by a donor that has the Delta CCR5 mutation. So since the Berlin patient, which he is popularly called, we have other individuals like the Dusseldorf patient, City of Hope patient, the New York woman, the London patient, and most recently, there is an individual that has been referred to as the Geneva patient that has displayed this profile of cure from HIV after undergoing stem cell therapy. And these different patients have provided a template for scientists to begin to understand what it means to be cured of HIV and what are the approaches that can be employed to achieve an HIV cure. But it's important to note that this is still something that we are hoping will happen in the future. So HIV is primarily a disease of the lymphatic system, and the lymph nodes are the major sites of active virus replication. So it's best to study immune responses at the site of initiation, which in all cases is within the lymph nodes or lymphoid tissue. And then within the lymph node, we have this area called the Germinal Center, where we have immune cells that interact closely. And the most prominent cells that interact within the germinal centers are the B cells and the T follicular helper cells. So the B cells are the cells that make antibodies, and the T follicular helper cells are a very important CD4 T cell subset that closely interact with B cells to guide the antibody maturation of B cells. So imagine this as being a marriage of TFH and B cells, and then their interactions within the germinal center actually helps the B cells to make very good affinity matured antibodies. The interesting thing about T follicular helper cells is that while these cells are good helper cells to B cells and CD8 T cells, these cells also have been shown to serve as a major compartment for HIV infection and replication and HIV production. So in our group, we are very, very interested in understanding T follicular helper cell function as helpers of B cells, but as also cells that serve as cellular reservoirs for HIV. And this is from some of the work that we have conducted within peripheral blood, we have shown, similar to other groups, that TFH responses are very important for HIV control. So in this data we published in 2018 in Journal of Virology, we show a correlation between a specific T follicular helper cell, which I'll be referring to as TFH, the TFH1, subset of TFH. We show a correlation between the frequencies of TFH in early acute HIV infection and the development of P24 IgG antibody titers later on after 1 year of infection. Moreover, the individuals that had the highest amount of this P24 IgG are the individuals that have the lower set point viral load. And the mechanisms for this for the P24 IgG to maintain HIV control in the absence of antiretroviral therapy has been clearly defined by many groups, including this publication by TGM. They have quite a few publications that explain how the mechanisms by which P24 IgG is able to control HIV viremia. And our studies were conducted in chronic in acute untreated HIV infection, but other studies in treated individuals and also in controllers of HIV have been shown that this TFH function is important for the development of HIV-specific antibody responses. But these studies were conducted in peripheral blood, and as I mentioned, the best place to study T follicular helper cell function would be within the lymph nodes, which would be the focus of my presentation today. And also importantly, we will be studying individuals that start antiretroviral therapy to understand the development of T follicular helper cell responses in these individuals. So to do that, we have this very unique cohort that is called the FRESH cohort. The FRESH stands for Females Rising Through Education, Support, and Health. This is a cohort that was established in Durban, South Africa in 2013, 2014 by some of our colleagues. And the characteristics of this cohort have been well described by Krista Dong and colleagues in 2017. But suffice to say, in this cohort, young women who are aged between 18 to 23 are recruited because they are at high risk of HIV, of acquiring HIV, but at the time they are recruited, they are living free from HIV. And the goal of the cohort is to give them information on HIV prevention and also equip them with important skills to actually benefit their lives. And they visit the center twice weekly. So because of their constant frequent visits to the center, they are they undergo rapid test for HIV. And for those that are found to have a positive test, then these individuals are then recruited into another arm of the cohort where samples are collected for other studies based on their consent. So as of early this year of July, we have enrolled almost 3,000 individuals into the cohort, and they have discontinued over time for those that are HIV uninfected. And so far, up to 96 acute infections have been identified, and these individuals are placed on ART immediately. I must mention that the first 14 individuals that were identified with a positive HIV test in 2014 or thereabout, those individuals were not placed on immediate antiretroviral therapy because at the time, the guidelines in South Africa only allowed for the initiation of antiretroviral therapy after a certain threshold of CD4 T cell counts. But that guideline has since changed, and everyone that has been detected with HIV infection has been placed on immediate antiretroviral therapy, some even as much as 1 day since the last negative test. So this allows us to identify acute HIV infection, to conduct studies on acute HIV infection, and some of the individuals consent to donate lymph nodes and other samples that we then also use for our subsequent studies. So I'd like to tell you a bit about our study design. So for the presentation today, I'll be telling you about a study where we recruited 64 participants. Some were from this FRESH cohort. We had 13 individuals that didn't have a positive HIV test. And then we also had 51 individuals. Some are from the FRESH cohort, but then we also recruited individuals that are late treated from other acute HIV infection cohorts in Durban, South Africa. The early treated individuals were those that started antiretroviral therapy, either in phoebic stage 1, or we also have 5 that started antiretroviral therapy in phoebic stage 3 to 5. And then the late treated individuals started antiretroviral therapy much later in infection, mostly in late acute HIV infection. And as I mentioned, our key focus was to understand the quality of T follicular helper cell responses, to understand whether there's antigen persistence in early treated lymph nodes, and also to evaluate what mechanisms are in play at either restricting persistence or in promoting persistence. So I'd like to give you a profile of the early treated individuals. So these are the phoebic stage 1. These are the 14 individuals that started antiretroviral therapy in phoebic stage 1. And this is the time point at which the lymph nodes were donated for this study. So we have a group of individuals that donated their lymph nodes within the first month of infection. And then we also have individuals that have been on antiretroviral therapy for up to 1 year, or even as most as 4.5 years. So this we have this particular participant have been on antiretroviral therapy for more than 4 years before the lymph node was collected. And as I mentioned, our first focus was on understanding T follicular helper cell responses within the lymph nodes of these individuals that start therapy very, very early. So the first question was whether germinal centers are actually present within these lymph nodes. And remember, I said germinal centers is that place, that structure within the lymph node where TFH cells and B cells interact. So for our imaging, so these tissues are fixed and paraffin embedded, and then we'll make 10 sections that we stain with antibodies. So the BCL6 antibody is used to identify active germinal centers, and then we identify T follicular helper cells by the expression of PD-1 or CD4, as has been well described in literature. So these are sample images. From the image on the left is from an individual who initiated therapy in phoebic stage 1. And the one on the right is for an individual who initiated therapy much later, I think phoebic stage 5. Excuse me. And interesting, as I mentioned, we observed that these individuals indeed had active germinal centers, and we could identify T follicular helper cells within the germinal centers. So we performed image cytometry to quantify the germinal centers. And from our quantification of our different subjects, we observed that germinal centers are induced and the sizes are higher in the early treated individuals when you compare to HIV unaffected individuals. But the magnitude of the germinal centers is much more significantly lower than the untreated infection. So the treated individuals, both the early and the late treated groups, had smaller germinal centers compared to untreated HIV infection. So from when we get the lymph nodes, we section the lymph node into 2. We use half for, we fix, and then we paraffin embed the half, which is what we use for our imaging studies. But then the other half of the lymph node is also disaggregated to obtain single cells that we use for other experiments such as flow cytometry. So in keeping with the phenotypic properties of T follicular helper cells, these germinal center T follicular helper cells have the highest expression of PD-1 and CXCR5 among CD4 T cells. And so here, these are representative images from HIV negative lymph node, and this is from an untreated lymph node where you can see this huge expansion of germinal center TFH cells that has been well described in literature. Interestingly, we also observed an induction of germinal center T follicular helper cells within the early treated and late treated groups, but the magnitude was much smaller than the untreated. And similar to the image or to the results that we obtained from the imaging studies, this is from flow cytometry studies, we observed the same kinetics or the same distribution of T follicular helper cells among the groups. So the expansion in the early treated is modest, and this is significantly lower than in untreated HIV infection. And this is particularly important because the high expansion of T follicular helper cells in untreated HIV infection has been associated with aberrant antibody responses. Excuse me. So we were also interested in investigating whether the T follicular helper cells are HIV specific. To do this, we have class 2 tetramers that we use to identify HIV specific cells. And so for the individuals that had this specific class 2 alleles, so we have tetramers to the DRB, 1301 and 1101 alleles, and individuals in our cohort that had these specific alleles were able to perform tetramer staining to quantify tetramer specific T follicular helper cells. And this representative image shows that indeed we were able to detect tetramer specific T follicular helper cells within the lymph node and within the peripheral blood. And because we only had few individuals that had the designated alleles, we couldn't perform tetramer staining on the whole cohort. But suffice to say, HIV specific Germinal Center T follicular helper cells were identified in this our study. As I mentioned, one of the primary functions of T follicular helper cells is to provide help to B cells in producing very good affinity matured antibodies. So we performed this co-culture assay to evaluate the function of T follicular helper cells in providing B cell help. And from our co-culture of T follicular helper cells with naive B cells, we measured the antibodies that were produced in the supernatant. And our results show that individuals that were treated very early were more efficient at inducing class switching of naive B cells to produce IgG. We also measured IL-21, which is a very important cytokine for TFH help to B cells. And individuals that were treated very, very early had higher expression of IL-21 as measured by digital droplet PCR. So I'll just conclude on this part, this first question that we asked, which is to say that we identified that germinal centers are induced in early treated individuals, as well as germinal center T follicular helper cell responses, but these responses are modest in comparison to untreated HIV infection. And importantly, these TFH responses in early treated individual have superior B cell helper function compared to TFH in untreated donors. Now we moved ahead to then answer our question about HIV persistence in these individuals that started antiretroviral therapy very, very early after infection, within the first few days after after acquiring HIV, and have been on treatment for up to 4 years for some. We used our imaging technology to identify the foci of HIV persistence within the lymph nodes. So for these images, GAG-P24 staining are in yellow, and then BCL6 staining are in green to identify germinal centers. So these are representative images from untreated, late treated, and phobic stage 3 treated individuals. And we can identify GAG-P24 staining, especially within the germinal centers, and also it's distributed in the extrafollicular areas for some tissues. Now, we focused on the early treated individuals because this is the focus of our study, and these are individuals that started treatment in phobic stage 1. And what we identified is a heterogeneity in the persistence. So you can see, moving into, zoom into the lymph nodes, we can identify lymph nodes that have quite a lot of that have a lot of GAG-P24 antigen. We also have some disaggregated germinal centers that have just very little P24. And then we also have germinal centers that don't even have any P24, all within the same lymph node in the early treated individuals. So focusing on our early treated donors, as I mentioned, we have heterogeneity in the distribution of P24 within the donors. Suffice to say, of the 14 early treated phobic stage 1 treated lymph nodes, only for two patients we didn't detect any GAG-P24, but for the other 12 we had different amounts of P24 depending on the germinal center. And to summarize the data for all our patients across the groups, there is persistence of GAG-P24 in phobic stage 1 individuals, but it is significantly lower when you look at phobic stage treated individuals or late treated individuals or untreated individuals. So there is persistence in early treated individuals, but the amount of persistence is significantly lower. We then also wanted to find out whether the persistence had to do with the duration on treatment. Remember, I said that our lymph nodes were not collected at the same time for all the phobic stage 1 treated individuals. So we then performed the correlation between GAG-P24 staining and the treatment duration, and there was no correlation, which means that P24 staining was not just dependent on the length of time that the individuals have been on treatment. Also, we observed that most of the GAG-P24 in most of the individuals was highly expressed within the germinal centers as compared to the extra follicular areas within the lymph nodes. Having observed a persistence of GAG-P24 protein, we also evaluated whether there's ongoing transcription within the lymph nodes in these early treated individuals. And to do that, we performed in situ hybridization to detect HIV RNA within these lymph nodes. And as you can visualize here, the HIV transcripts are in this green color. So these are dots in green color, but because of the concentration of the RNA, you can see it's almost like you have like these blobs, and then T follicular helper cells are stained in red here, and the nuclei are stained in blue. And what we observed here is that there is ongoing transcription in the lymph nodes of early treated individuals, but the magnitude of RNA, similar to the magnitude of P24 protein, is significantly lower, especially in all the early treated individuals, especially within the individuals that start antiretroviral therapy in phobic stage 1. So starting therapy very, very early in HIV infection is beneficial to reduce the antigens that persist, either as proteins or as RNA or transcripts within the lymph nodes. So we were also interested in understanding whether there are specific cellular subsets that actually harbor the greatest amount of the reservoir. And from our previous studies and in keeping with literature, we know that TFH cells are heterogeneous, and there are different subsets of TFH based on the expression of markers like CXCR3 and CCR6. So we sorted these cells that are expressing CXCR3 or not expressing CXCR3. And what we found was an enrichment of HIV mRNA within CXCR3 positive expressing cells. We also then performed a phenotypic characterization of the cell suspensions, looking for CXCR3 marker having stained with a 3 BNCC117 antibody to identify GP120 positive or GP120 expressing cells. And our results also show that CXCR3 positive TFH cells are the cells that harbor the greatest amount of GP120. So these CXCR3 expressing cells are enriched for the persistent HIV in early treated individuals. Moving on to the last aspect of my presentation, where we interrogated the mechanisms of HIV persistence or clearance from the lymph node sanctuary sites. So studies have shown that cell-to-cell spread or cell-to-cell transmission is one very important mode of HIV transmission, especially in tissue sanctuary sites. And cell-to-cell spread could be contributing to persistent virus replication in the tissue. So what we did was to then isolate viruses from, so these are chimeric viruses. So these are GAG proteins, chimeric viruses, which were generated using patient GAG protease that is isolated from patient samples and inserted in the same NL43 backbone to generate replication competent viruses. And so we have GAG from plasma, from lymph node, and from PBMCs, and then we perform cell-to-cell spread experiments to measure the efficiency of these cells at cell-to-cell spread. So what we observed was there was no differences in cell-to-cell spread between the plasma of the viruses from plasma, lymph node, or from PBMCs. But importantly, all these viruses were efficient at conducting cell-to-cell spread. We also sequenced the GAG that was isolated from patient samples, or that was amplified by PCR from patient samples. And we performed this deep sequencing to identify whether there were specific mutations to the viruses that will make it more efficient at cell-to-cell spread, or to have like specific characteristics. And the results we got from our sequencing was there were limited mutations, but especially specific patients. We had these 3 specific patients that had important mutations that have been identified as CTL-driven mutations. So these then guided us to begin to also study CD8 T cell, the role of CD8 T cells in promoting or in controlling persistent virus within the lymph node. And this part of the work, I didn't mention the previous part of the study for the viruses was led by a colleague called Dottie, whose picture I displayed. And this part of the work was led by a colleague called Funshaw. And so the first thing was then to identify CD8 T cells within the germinal centers. So here, BCL6 identifying germinal centers, and then here the red staining is for CD8. The orange is for GAG-P24, and you have the matched image. But importantly, you can see here that within the germinal center, we don't have a lot of infiltration of CD8 T cells. And this is a summary for all the images that we analyzed, and you can appreciate that the frequencies or the densities of CD8 T cells was higher outside the germinal centers than within the germinal centers. And then we also have data, which I'm not showing here, that also shows a correlation between infiltration within germinal centers and CXCR5 expression. So essentially, CXCR5 expression licenses CD8 T cells to be able to migrate into the germinal centers. If you can remember, the TFH cells have very high expression of CXCR5. And so we then quantified antigen-specific CD8 T cells that are expressing interferon gamma, and then we measured the expression of either CXCR5 or lack of expression of CXCR5, and we can see that most antigen-specific CD8 T cells actually don't express CXCR5, which shows that, which also ties to the fact that CD8 T cells are not licensed to migrate into the germinal centers to eliminate infected cells or persistent virus-infected cells. We also evaluated the function of CD8 T cells using a proliferation assay that is called the CFSE assay. And then we correlated the results of the proliferation assay which measures the proliferation in response to the HIV gag antigen. And we correlated that with the density of gag P24. And we observed that individuals that had higher functional proliferating CD8 T cells actually had lower density of gag P24, which seems to suggest that CD8 T cells may be playing a role in controlling persistence in early treated individuals. So, because CD8 T cells are not allowed to migrate into the germinal centers, then there's antigen persistence in the germinal centers. And we sought to investigate whether there is a solution to allowing CD8 T cells to be able to migrate into the GCs. So here we tested this recombinant TGF-beta antibody to whether it's able to increase CXCR5 expression on CD8 T cells. And as we can see here, stimulation with recombinant TGF-beta is able to induce CXCR5 expression on CD8 T cells. And we think that this might be an important solution to, especially in individuals that have persistent viremia, to allow clearance of virus-infected cells within germinal centers. So in conclusion, from this study, we observed that individuals that start antiretroviral therapy indeed still have a germinal center response, and they still have the T follicular helper cell response, but the response is modest in comparison to untreated individuals. And this modest response is actually beneficial to these individuals because we have data that shows that the TFH cells in early treated individuals are more functional than TFH cells in untreated individuals. Also, we showed that though these individuals start antiretroviral therapy very, very early, and some of them have been on treatment for more than 4 years, there is still persisting HIV gag P24 protein and transcript within the germinal centers. And we have identified an important marker, which is CXCR3, as a marker of TFH cells that harbor the greatest amount of persistent virus. And future studies can design interventions to target these specific cells in order to reduce persistent virus within the tissues. The virus between the lymph nodes is efficient at cell-to-cell spread transmission, similar to plasma and PBMC viruses. And we think that some of the reasons why there's persistent antigen within the germinal centers is due to the limitation of CD8 T cells from accessing these immune sanctuary sites. And we profile one of the solutions to be increasing CXCR5 expression on CD8 T cells, and that can license CD8 T cells to enter the germinal centers and eliminate persistent virus. So poor migration of CD8 T cells contributes to persistence, and we can reverse this by licensing CD8 T cells to enter the germinal centers by increasing the expression of CXCR5. I'd like to acknowledge Professor Zazan Vlovu, in whose lab I performed most of these studies together with my colleagues. My mentor is Prof. Tumbi Ndungu. I would like to acknowledge my colleagues that have contributed to this work, and importantly, our funders and study participants. Thank you.
Wow, Dr. Baiyegunhi, thank you so much for your talk on the latest research in HIV. It's really amazing to see that you actually have human subjects for your studies, making your findings all the more applicable to how our human bodies work in health and disease, and also potential avenues of therapy. So I open up the floor to questions, and we have one. Somebody had asked for mechanisms of HIV, understanding HIV persistence during ART, which you talked about in the beginning in your introduction. Can you delve deeper into the concept of tissue sanctuary sites in the context of HIV persistence? For instance, how do these sites contribute to viral rebound despite early ART initiation?
Yeah, thank you. That's a very important question. And I think that that question was typed into the chat before the end of my presentation, because I think the focus of my presentation was trying to explain that these tissue sanctuary sites actually are immune privileged sites that actually exist. And because CD8 T cells, which are the killer cells, are not licensed to enter these sanctuary sites, then there can be a persistence of virus within the site. And because of this persistent virus, which we also show that there's some transcription going on, it's possible that this persistent virus could also contribute to rebound in individuals once they stop antiretroviral therapy. This persistent virus may contribute to the virus that will rebound.
Got it. We have two more questions for you, so lots of interest in you. This one may or may not be similar. I'm sorry, my background is not in HIV research, so I'm just going to read it exactly as it is in our chat box. Dr. Baiyegunhi, thank you so much for a great talk, especially seeing that you are joining us from South Africa. You mentioned that virus rebound occurs even in individuals initiated on ART during the phoebic stage 1 HIV infection. Could you provide insights into why this rapid rebound occurs despite the lower numbers of latently infected cells in the peripheral blood?
Yeah, so the thing about, I think that's the interesting thing. All it takes is just one virus to start the rebound. So once one virus is actively replicating, then we know that HIV multiplies very, very quickly. So yeah, as long as there is that one virus that is viable and capable of replicating, when individuals stop taking antiretroviral therapy and that virus starts replicating, then we have high viremia. But I think many of the individuals that have been described as being cured, scientists have conducted studies to try to detect a viable virus, and they've not been able to detect intact viruses that are viable for to replicate, and these individuals have remained suppressed over this long period of time. So so I guess it depends on if there's a virus that can replicate, then rebound will occur. But then there are depending on the individuals, there are certain immune responses that will be able to, suppress this replication. So we have different kinetics of rebound. So some individuals rebound faster than others.
Got it. Wonderful. We have one more question for you, kind of a thought question for the future, it sounds like, so I'm saving it for the last part. So this individual asked, given your research focus on understanding HIV reservoir persistence, what do you think are promising new or continued avenues for developing novel strategies to target these tissue sanctuary sites and achieve HIV eradication or cure? So $1 million question for you, Dr. Baiyegunhi.
I agree, that's $1 million question. So we have many ongoing studies. We have studies where we are characterizing the proviral reservoir to identify individuals that have either high or lower amounts of the reservoir. And then we are then grouping these individuals based on the magnitude of the reservoir and trying to understand the mechanisms that contributes to a high or low reservoir and how this can be targeted. So some of the approaches have to do with metabolic determinants, which these are maybe like metabolic modulators, drugs that can modulate cellular metabolism or cellular metabolic based pathways. So these are some of the kind of studies that we are undertaking to identify what are the pathways that are contributing to the magnitude or the viability of the reservoir, is the ability of the reservoir to rebound, and how can then we introduce drugs into the system that can suppress this rebound viremia. So we have quite a lot of studies that are ongoing in our lab to try to answer that question, which is also important for many researchers in the HIV research field.
Thank you very much. So Dr. Baiyegunhi, again, we want to extend our thanks, and we certainly hope to have you here again in the future to give an update on your exciting research. And with that, I want to also thank all of our attendees. And if you have any questions, please reach out to us. And I'm sure Dr. Baiyegunhi will be happy to share more of her research findings with you.
Thank you for having me. It's been great. And thank you to all those that attended the webinar.
It is our pleasure. All the best.
Thank you.